Great, Ron. I think you could go ahead whenever you feel like it. So give a little intro.
Well, sure. I'm happy to make a comment. So, I mean, first of all, I think I just want to say something quite general, how nice it is to be getting together in this setting. And I found after the last session we had, it was very nice almost immediately to go back and look at some of the video that May had recorded. So you could sort of remind yourself of some of the discussion that had just taken place, and you didn't have to remember everything. You could go and think about it more. So that's to say, I think this is really important that we talk this way. And I want to make just a couple of very general points. And I want to build them around the ideas of stem cells, equivalence groups, and organizers. These ideas are central to our field. And just to sort of not to sound too much like a graduate class, but a stem cell is a member of an equivalence group that can adopt a series of potential fates under organizer-based patterning signals. That's just a statement of whole decades of developmental biology, starting with Mangold and Spearman. So for those of us interested in the nervous system, we can immediately think of several examples, and I'm just going to list them now just to stimulate your thought. The Drosophila retina is an equivalence group. The vertebrate spinal cord is an equivalence group. The mammalian cerebral cortex is an equivalence group. But of course, omnicellular, ecellular, so it's not surprising that all of contemporary biology and medicine is deeply vested in the details of cell lineages. And our knowledge of these types of cellular dynamics is currently being transformed by many new tools that allow us to perturb and define the relations between cell types. And many of these use pluripotent cells as a model system. So as we better define the equivalent core mechanisms in the stem cell compartment, we can increasingly define the nature of variation between cells. And that I think is what makes the timing of this group getting together so opportune. It's such a great time because we all come at this, it seems, with slightly different perspectives. And in our first meeting, we heard fascinating talks on different aspects of developmental diversity using pluripotent cells. And today we'll hear 2 more talks on this topic, again, slightly different, very interestingly different. So I want to close just by making a very important point. All of contemporary human genetics teaches us that genes matter, but how they act to cause different phenotypes is a huge problem. And what I'm going to propose to you is that the work that we are doing is not just a sort of sideline, it's a central feature of developing a new understanding of biological systems. And this variation is not some technical artifact, it's the way biological systems are built. And so to emphasize how important this is, I just want to use this sort of slightly catchy phrase, the dice are loaded. We know the dice are loaded. Now the question is to use these concepts of stem cells and equivalence groups and organizers to gain a biological and mathematical and clinical granularity as to the consequence of this variation. That's the task in front of us. And I think if we can share these ideas with each other, I'm very interested to see how our discussions develop, because they're bigger than any one of us can easily summarize in 2 or 3 minutes anyway. So I'm very excited by the discussions, and I just wanted to broaden it out slightly using these ideas that are core in developmental biology, and make sure that we enjoy thinking this thing through at many different levels. So I'll leave it at that. Thank you very much for asking me.
Thanks, Ron.